about his
স্যার
size. বলো
একজন আমাকে মেসেজ করছে।
To identify biomolecules. First of all, in order to become biomolecules, some specific conditions are required to be satisfied. First of all, it should be suppose whenever it is separated out, there are two. It is a acid fractional pool, other is the non-acid fractional pool. So whenever they are separated, if they are separated acid soluble pool or acid insoluble fraction, two types. Fraction or acid look, either they may be collected in the acid soluble portion or in the acid insoluble fractions. So that those who are collected in the acid soluble fractions, there should be the and the next one is, this is the first point of type. Second one, molecular weight. Generally, if it is an organic compound, biomolecules, it should be এখানে একটা তো ভাগ করে নি, একটা ভাগ করতে হবে। Biomicromolecules and biomacromolecules. The first point of difference. If they are from the biomacromolecules, the molecular weight (18 to 800 Dalton). So, in case of biomacromolecules, their molecular weights lie from 18 to 800 Dalton, and biomacromolecules should be macromolecules found in the acid soluble fraction. Regarding one most important thing regarding the biomacromolecules are carbohydrates, the protein, nucleic acids. In terms of its molecular weight, but lipid, in terms of its molecular weight, it is found to be biomicromolecules. Biomicromolecules are found to be present in acid soluble, found in
স্যার স্যার লিপিডের জায়গাটা কি?
Lipid is most interesting thing. It shows regarding the molecular weight, it is under biomacromolecules, but it is also found in acid insoluble pool like the macromolecules. বুঝতে পেরেছি কি বলেছি? A peculiar characteristic it shows regarding the molecular weight, it is under the biomacromolecules, and regarding the how that is found in acid soluble or insoluble pool, it is found with acid insoluble pool like that of the biomacromolecules. কথা বুঝতে পেরেছিস? In terms of molecular weight, it is under biomacromolecules, but while collection of them in the acid soluble or insoluble, it is found to be in the acid insoluble fraction.
insoluble fraction.
Biomacromolecules are found in acid soluble. Found. কোথায় পাওয়া যায়? When there to solubility test is done, they are found in acid soluble fraction, micro. A macro is found in acid insoluble fraction. বুঝতে পেরেছো?
Yes, sir.
Now regarding one exception is there only. This is regarding the lipids actually. You know. So now we have to discuss with them.
স্যার
বলো।
স্যার লিপিডের রোলটা এখানে বুঝতে পারিনি।
Interesting about this macromolecules, one thing you know, what I said, that if you have to classify molecules in terms of molecular weight, so molecular weight 18 to 800 Dalton, it should be biomacromolecules. If it is more than 800 Dalton, it should be biomacromolecules. Clear?
Yes, sir.
Now regarding lipid, if you consider it as a defining property that yes, if it is a biomacromolecule, it should be within 18 to 800. If it is a biomacromolecule, it should be more than that. Clear? Now, lipid is molecular is found in the range of 18 to 800 Dalton. Technically, it should be biomacromolecules. Clear?
Yes, sir.
Is it clear up to this? Technically, it should be under biomacromolecules because its molecular rate ranges in between 18 to 800 Dalton. Now, regarding its collection, interesting thing is they are found to be present in acid insoluble fraction. Although its molecular weight is in the range of 18 to 800 Dalton, but it is found to be present in acid insoluble fraction, similar to that of biomacromolecules. বুঝতে পেরেছিস?
Yes, sir.
So in one point of view, technically it should be biomacromolecules, but as it is found in the acid insoluble fraction like that of other biomacromolecules, we have to study lipid along with other biomacromolecules. বুঝে গেছে?
Yes, sir. স্যার অ্যাসিড ইনসoluble পুলটা কি?
হ্যাঁ
স্যার পুল যে লিখেছো পুল
মানে হচ্ছে অ্যাসিড ইনসoluble পুল মানে হচ্ছে এখানে এটা লিখতে পারি আমি একই জিনিস অ্যাকচুয়ালি whenever that in a test tube if you take the macromolecules, then if they are found soluble in acid, then they should be under the acid soluble pool. But if they are not there, if they are found to be sedimented, they should be in the acid insoluble pool. বুঝতে পেরেছিস?
হ্যাঁ স্যার।
এই অবধি ক্লিয়ার হয়েছে? দেখেন ভালো করে। We have to proceed towards each type now. First of all, protein. All are amino acids. Linked by peptide bonds, and you can call it a protein. So what is protein? Protein is a macromolecules which is made of a number of acids linked by peptide bonds. I think you have idea about the peptide bonds. I discussed it earlier, right? So each one is amino acid, but amino acid is not a protein. Although building block of protein is amino acid, but amino acid is not a protein. Can you tell me why? Although building block of protein is amino acid, but amino acids are not considered as protein. Why? In order to be considered as protein, at least one peptide bond is required to be present. বুঝতে পেরেছিস? In order to be considered as amino acid, at least one peptide bond is required to be present, which is not present in a single amino acid. A single amino acid may be RCHNH2COOH. This is actually the amino acid. Having one amino group, one carboxylic group. Then it should be amino acid is an organic acid. At least having at least one amino group, amino acid is an organic acid. Having at least one carboxylic group, having at least one amino group and one carboxylic group. Then you can call it, equally you can show it like this also. Here, the simplest value of R equal to H. So simplest amino acid should be it is called glycine. বুঝতে পেরেছিস?
স্যার অ্যামাইনো অ্যাসিড প্রোটিন কেন না বললে?
Because in order to be considered as a protein, at least one peptide bond is required to be present. But you know, peptide is found in between two amino acids. So in a single amino acid, how a peptide bond can be present? বুঝতে পেরেছিস?
Yes, sir.
That's why it is not considered as an amino acid and a protein. So this is the simplest amino acid. So CH2NH2COOH, and this is called the glycine, the simplest amino acids. Then comes to the different types of amino acids. We are going to classify it. First of all, you need to understand the how to define amino acids. Next, একটু মুছে দিয়ে যাবে একটা মিনিট, দুইটা মিনিট একটু আসছে মেয়ে, জাস্ট মিনিট প্লিজ, একটু দেখে নাও এটা। Just 2 minutes. The RCH2NH2COOH is the simplest amino acid because the simplest value of R is H. Now, next the classification. একটু মুছে দিই জায়গাটা। is sometimes found in this form also. This is called deuterium. Amino acid at a particular pH, amino acids are found to be present in deuterium form. Look here. When release one proton, it may be the acidic form or negative form. It may be by adding one, by getting one proton, it may be in the positive form. But when both positive and negative charge, they contain both the charges. When amino acid with both the charges called deuterium, so net charge equal to 0. Look. Is clear what is deuterium? Actually, deuterium is a hybrid molecule having both positive and negative charges. Deuterium refers to a hybrid molecule having both positive and negative charges, so that net charge is 0. So that net charge is 0. And at what pH an amino acid is found to be present in deuterium form? At isoelectric pH. It varies amino acid to amino acid. Clear? দেখ বুঝতে পেরেছিস কিনা, এটা খুব ইম্পর্টেন্ট কিন্তু deuteriumটা।
Sir, isoelectric base ব্যাপারে কি বলছেন?
Isoelectric the pH at which an amino acid is found to be present in form.
আইসোইলেকট্রিক পিএইচটা কি বলেছো?
কথাটা শুনতে হবে তো আমাকে, আমার কথা না শুনলে আমি তো শুরু করলে মাঝখানে আবার বলে কে কোনটা শুনবি তুই, সেটাই বল। The pH at which an amino acid is found in deuterium form is called isoelectric pH of that amino acid. Suppose leucine, one amino acid is called leucine. At pH 6, leucine is found in deuterium form. So for leucine, the isoelectric pH is pH 6. So it varies amino acid to amino acid. What should be one's deuterium form? দেখ বুঝতে পেরেছিস কিনা? দেখো বুঝতে পেরেছিস কিনা আগে দেখ।
স্যার জেপিএইচ জুইটারিয়ান ফর্ম এটাকে বলে না আইসোইলেকট্রিক?
হ্যাঁ হ্যাঁ, যে পিএইচ এর deuteriumটা ফর্ম হয় তাকে আমরা বলছি হচ্ছে আইসোইলেকট্রিক পিএইচ।
ঠিক আছে স্যার।
বুঝতে জিনিসটা?
ইয়েস স্যার।
Now, deuterium clear, isoelectric pH clear. Now we have to classify amino acids based on. মুছে দিই একটু। Now the classification of amino acid. You can classify amino acid into acidic, alkaline and neutral type. Acidic হচ্ছে দেখো ওটা amino group and carboxylic group.
স্যার অ্যামাইনো অ্যাসিড অ্যালকালিন কেমন করে হতে পারে?
হ্যাঁ,
স্যার অ্যামাইনো অ্যাসিড অ্যালকালিন কেমন করে হতে পারে?
এটা বেসিক amino acid বলে এটাকে, হতে পারে।
স্যার অ্যাসিড হয়ে বেস হয়।
হ্যাঁ, বেসিক amino acid হতে পারে, কেন হবে না? রাইন অর বেসিক বলবো, আমি দেখাচ্ছি দাঁড়াও। ফার্স্ট অফ অল অ্যাসিডিক, look one thing, a different color thing. It is all normal. Then CH2, CH2, COOH. Look at this diagram. Here number of carboxylic group 2, number of amino group 1. That means it is an acidic amino acid. Example, so it is acidic amino acid. The number of amino group is more than the number of carboxylic group. Clear? আরেকটা দেই, this is the second one. যদি আমি আরো ডিটেলসে দেখাই, দেখানো এরকম this should be a form তোমাকে বুঝতে সুবিধা হবে। এটা হচ্ছে aspartic acid. So why these two are called acidic amino acid? Clear? Clear? এবার দেখ অ্যালকালাইন অ্যামাইনো অ্যাসিড। সেটা বলা হয়েছে। দেখো তিনটা মিথাইল গ্রুপ দিলাম, so you can call it lysine. Basic amino acid. The number of amino group here more than the number of carboxylic group. That's why it is basic. In case of neutral, it is one neutral amino acid.
স্যার স্যার ওই লাইসিনের স্ট্রাকচারটার এটা কি এটাকে
হ্যাঁ কি হয়েছে বলো।
লাস্টের নিচেরটা এটা
হ্যাঁ নেক্সট টু বুঝলি বুঝলি
জি স্যার।
আমার ধরো আর একটা কমন অ্যামাইনো অ্যাসিড নিউট্রাল অ্যামাইনো অ্যাসিড আছে, যেমন ধরো অ্যালানিন, মিথাইল গ্রুপ কন্টেনিং অ্যামাইনো অ্যাসিড। কিরকম এটা? এটাও নিউট্রাল অ্যামাইনো অ্যাসিড। আর ধরো হাইড্রোক্সিল গ্রুপ কন্টেনিং অ্যামাইনো অ্যাসিড, called hydroxyl group containing amino acid. It's called serine.
স্যার হাইড্রোক্সিল গ্রুপ কন্টেনিং অ্যামাইনো অ্যাসিড কি এটা কি নিউট্রালের পার্ট?
হ্যাঁ দেখি হবে, বলো নিউট্রাল হবে কি মনে হয়? Can it be a neutral amino acid? What do you think? বল এটা কি নিউট্রাল অ্যামাইনো অ্যাসিড হবে? হ্যাঁ
হ্যাঁ স্যার।
অবশ্যই, because you know number of amino group, whenever it is equal to the number of carboxylic group, it should be a neutral amino acid. So serine is a neutral amino acid. So now question comes, name one hydroxyl group containing amino acid. The answer should be serine. আমি একটা এক্সাম্পল দিচ্ছি এখন, অ্যারোমেটিক অ্যামাইনো অ্যাসিড, ফিনাইলঅ্যালানিন। Aromatic having benzene ring. So it is an aromatic amino acid containing a straight side chain bearing carboxylic group and amino group and a cyclic ring. Actually, aromatic amino acids are those amino acids whose R group contains a cyclic ring. Then you can call it aromatic amino acid. Sulfur containing amino acid methionine and cysteine. একটা ইমেল পরীক্ষা আসে, methionine, cysteine. They are the sulfur containing amino acid. Suppose in case of cysteine appears like this. This is cysteine. So in this way we can classify different amino acids. দেখা অবধি বুঝেছো কিনা?
স্যার
বলো।
সালফার কন্টেনিং অ্যামাইনো অ্যাসিডের পরেরটা কি লিখেছো?
একটা হচ্ছে মিথায়োনিন আর একটা হচ্ছে সিস্টিন। আমরা মিথায়োনিন স্ট্রাকচার দেখিয়েছি, যদি সিস্টিন মিথায়োনিনটা যদি দেখাই কিরকম হবে, This is the methionine. বুঝতে পেরেছিস স্ট্রাকচার সম্পর্কে আইডিয়া হয়েছে? তোমাদের বইতে বোধহয় এতগুলো নেই, আমি কয়েকটা বেশি দিয়ে দিয়েছি যাতে বুঝতে সুবিধা হয় তোমার। ক্লিয়ার?
Yes, sir.
আফটার দ্যাট মুছে দেবো একটু। মুছে দিই, হয়ে যেত সবার?
হ্যাঁ স্যার।
Essential, semi-essential, non-essential amino acid. You know, the amino acids which are not synthesized in our body but required for our nutrition, so they are to be taken from outside through diet, called essential amino acids. The amino acids which are essential for our nutrition but are not synthesized inside our body, as they are to be obtained through food, called essential amino acids. There are 8. Methionine, valine, tryptophan, threonine. These are essential amino acids which are not synthesized in our body. Semi-essentials, arginine and histidine. Why they are called semi-essential? This is because these amino acids are essential in infantile stage. In adult stage, they become non-essential. That means in the infants, body of infants, arginine and histidine cannot be synthesized. So these two amino acids are required to be taken through diet. But in the adult stage, these arginine and histidine are found to be synthesized in the body. So in adult form, both these amino acids are considered as non-essential. বুঝতে পেরেছি জিনিসটা এবার? The difference between essential and non-essential. And why it is called semi-essential? The other amino acids, out of 20, 8 are 1, 2, 3, 4, 5, 6, 7, 8. essential, 2 semi-essential, and other 12 should be non-essential. Like glycine, cysteine. Glycine, alanine, serine, tyrosine, glutamic acid, aspartic acid, cysteine, proline. Glutamic acid. Glutamine and asparagine. Asparagine. So these are the question of 20 amino acids which are required in our body. Clear?
স্যার এস পার টিকেট নিচেরটা কি?
Aspartic নিচে আছে cysteine. তারপরে হচ্ছে প্রোলিন, তারপর হচ্ছে গ্লুটামাইন, লাস্টে অ্যাসপারজিন।
স্যার 5 নাম্বারটি কি গ্লুটামিক্স?
গ্লুটামাইন, GLUTAMINE। আচ্ছা, 10টা হচ্ছে অ্যাসপারজিন, SPERGINE।
না না স্যার, ও 5 নম্বরটা জিজ্ঞাসা
স্যার 5
করেছিল।
বলছি, 5 নাম্বার।
এটা লাইসিন,
না,
নন এসেনশিয়াল।
non-essential এর।
গ্লুটামিক অ্যাসিড, গ্লুটামিক অ্যাসিড।
স্যার স্যার তুমি নন এসেনশিয়াল বলেছিলে রাউন্ড অফ 20, এটার এসেনশিয়াল আর আদার 12 আর নন এসেনশিয়াল, তাহলে তো স্যার সেমি এসেনশিয়ালটাও কাউন্ট হচ্ছে এর ভিতরে।
আউট অফ 20 দেখো মিলে 20 হয়নি, 20 হচ্ছে না দেখো তো।
স্যার 20 তো হচ্ছে।
হ্যাঁ, these two are semi-essential in child. Why they are called semi-essential? Because they are found to be essential in childhood, but non-essential in the adulthood. বুঝতে পেরেছিস এবার?
হ্যাঁ স্যার।
In adult stage, you will get 12 non-essential, and in infantile stage, you will get 10 essential. বুঝেছো এবার?
Yes.
These two are under a separate group called semi-essential. Clear?
Yes, sir.
মুছে দিবা। কি হয়ে গেছে তো? এর একটা প্রশ্ন বলতে কাকে জিজ্ঞেস করবো? অরিত্র বলো, how many amino acids are found in nature? How many amino acids are found in nature? How many amino acids are found in nature? হ্যাঁ, অপরজিৎ বলো।
Sir, 20.
বলো অপরজিৎ।
Sir, 500.
Not 500 actually, it is almost slightly above 300. More than 300 amino acids are found in nature. More than 300 amino acids are found in nature, but only 20 amino acids are found to form different proteins in the body of living organisms. মুছিদি সবটা? Next comes to the আবার দেখিয়ে দিই। আমি দুটো গ্লাইসিন নিচ্ছি। Both are glycine molecule. Peptide bond. Now this molecule is called dipeptide. So peptide bond is a covalent bond formed between the carboxylic group of one amino acid and amino group of the adjacent amino acid with the liberation of one molecule of water. Enzyme that catalyzes peptide bond formation is peptidyl transferase, and it is a ribozyme. Ribozymes are not protein in nature, actually RNA with catalytic role called ribozyme. So this is one peptide bond, how it is formed and how to define it. A covalent bond formed between the carboxylic group of one amino acid and the amino group of the adjacent amino acid with the liberation of one molecule of water. And this bond formation is facilitated by catalyzed by peptidyl transferase enzyme, which is actually ribozyme. So in this way, the formation of protein is initiated. Remember these things? In order to be considered as protein, at least one peptide bond is required. So it is the simplest protein, dipeptide. Dipeptide is a protein which is formed by the union of two amino acids. It's called dipeptide. Dipeptide not for the number of peptide bonds, but the number of amino acids involved to form the protein. How many amino acids are involved here? Two amino acids are involved. So a dipeptide molecule contains two amino acids actually. Or in other words, we can say two amino acids are linked with each other to form a peptide bond. Clear?
Yes, sir.
আমি আগে বলছিলাম না এটা আগে বলছিলাম না মনে হচ্ছে now the classification of protein. Primary protein, secondary protein, tertiary protein. দেখো বুঝতে পেরেছি কিনা দেখা যায় কিনা সবগুলো UUARTER NARY. Primary protein only peptide bonds are present. In primary protein only peptide bonds are found. In secondary protein so here we get then you can call it secondary protein. Secondary protein is of two types actually. One is alpha helix, this is the alpha helix type. Other is beta pleated type. And in the beta pleated type, what happens you see, this is one primary protein. This is the other primary protein. Suppose this is the third one. Maybe this is the fourth one. All are primary protein. This is primary protein, this is primary protein, this is primary, but they are linked. They are linked by hydrogen bonds here. Linked by hydrogen bonds. একটু বড় করে একটা ভালো দাঁড়া একটু ডিটেইলস করছি সুবিধা হবে দেখো এটা একটা প্রাইমারি প্রোটিন hydrogen bond so this is a clear now? These are both are secondary protein. Clear? Tertiary protein appear like this. So you see, beside the peptide bond, and hydrogen bond, disulfide bonds are also found. Then you can call it tertiary protein. Here beside hydrogen bond and a peptide bond, disulfide bonds are also present. Are you getting me? Then you can call it tertiary protein. Any enzyme, any protein, if it is to act as an enzyme, it is to attain tertiary structure. Most important thing. And on heating, enzymes deactivated because on heating, disulfide bonds are broken down. So is a tertiary protein clear? Beside hydrogen bond and peptide bond, disulfide bonds are also present. এটা করে নাও। bond or hydrogen bond, then it is called quaternary protein. When two or more tertiary proteins are linked by either hydrogen bond or ionic bond, then it is called quaternary protein.
Hydrogen bond or
Ionic bond. Quaternary protein. Clear?
Yes, sir.
yes, sir.
Okay. In hemoglobin, globin protein is made of 4 polypeptide chains. 2 alpha polypeptide and 2 beta polypeptide chains are linked to form the hemoglobin, and hence hemoglobin is considered as a tertiary protein, quaternary protein. Hemoglobin, the globin protein globin protein in hemoglobin consists of 2 alpha polypeptide chain and 2 beta polypeptide chain. And each polypeptide chain here, each alpha polypeptide, each beta polypeptide is a tertiary protein. And whenever they are linked by hydrogen bond, then it is then a quaternary protein is formed, which is called globin protein. Is clear? The globin protein is made of 4 polypeptide chains. Each polypeptide chain is a tertiary structure, and whenever they are linked to each other, a quaternary structure is formed. That's why it is called globin protein is an example of quaternary protein. The secondary protein, beta pleated protein. Beta pleated protein is of 2 types. One is parallel beta sheet, other is anti-parallel beta sheet. Parallel beta sheet and anti-parallel beta sheet. Parallel beta sheet is made of, actually example diye dao, beta carotene, clear? Beta carotene is the beta parallel sheet, parallel beta sheet. Hello? Parallel beta sheet, beta carotene, anti-parallel beta sheet, fibrin protein of silk. Sometimes this question coming in the entrance. So protein is completed. হ্যাঁ বলো।
স্যার গ্লোবিন প্রোটিনের মধ্যে 12 আর 2 বিটা কি থাকতে লাগবে না একটা থাকলে হবে?
না, if it is a hemoglobin. In case of myoglobin, it is different. But if hemoglobin is to be formed, then in adult hemoglobin, which is HbA type, obviously there should be 2 alpha and 2 beta chains. Fetal hemoglobin may be different. আমার কথা বুঝতে পেরেছিস?
হ্যাঁ স্যার।
So this is all about the structure of protein. আমাদের প্রোটিনটা শেষ হয়ে গেল আজকে। আমাদের বাকি থাকলো লিপিড, then comes to the carbohydrate and then the nucleic acid, and after the enzyme, then the chapter is completed. That's all for today. আজকে আমরা এখানে শেষ করছি। Next day we have to start with the then carbohydrate, okay, and then lipid. Carbohydrate and lipid complete by the next day. হ্যাঁ বলো।
স্যার তুমি বলছিলে কেপি পড়াবে বলছো।
কার্বোহাইড্রেট ফ্যাট শেষ করে দেবো, দেখি যদি নিউক্লিক অ্যাসিড শেষ করা যায়। Okay, that's all for today.